This works. Okay. That's a bit in the way, isn't it? Okay. So done that. Oops. Okay. Let's look at the group data from the assignment. So everyone's data averaged together, which when you do that, the results are always cleaner than the individual data. So here's reaction time mean, and we have our typical stimulus response compatibility effect. So I forgot to add pictures to this, but these two are the compatible responses. So that's left stimulus, left response. So that's the easy trial in that block. And then the other easy trial is right stimulus, right response. So that's one block when it's ipsilateral. And when we do that, you have relatively short reaction times, which makes sense. These two show the contralateral block. So this trial, let's see, that's a right stimulus, and you're making a left response. So it's the opposites, and then over there would be the other opposite contralateral mapping. So you're slower, nice robust effect across everyone. I'm trying to remember if all the individual data showed, I think all the individual data was pretty good for this. Reaction time standard deviation at the individual level was a bit messy or noisy. Not everyone had a really nice kind of compatibility effect X. But when we average across 9 of y'all, we do see a fairly good compatibility effect in standard deviation. So the compatible block is less variable, and the incompatible block or the contralateral block is more variable. One thing I didn't ask y'all to calculate was how many errors did you make, or how many incorrect trials were there? And the reason I didn't is because in the past, I've seen that it's very variable. And even in your data set, there was at least one participant who made no mistakes. Like on both blocks, all the trials always pressed the right key. And then there were some participants who made more mistakes. So you typically don't see this come out very well at the individual level, but at the group level, we can see a nice kind of compatibility effect. So these are the ipsilateral trials. Those are the contralateral trials. And there's not a ton of errors. So 0.05, that's proportion. So multiply by 100 to get percent. So that's 5%. So you can see, I guess across everyone, in the contralateral block, you make a mistake, let's say 4% of the time. And when it's ipsilateral, when it's easy, you make, well, I guess there are no errors here, or half of 1% over here. So that usually works out in the group data, but not at the individual level. So that's why I didn't ask y'all to do that. Here's our reaction time distributions, and I'm bringing this up because our last module, so not this one, but the next one, we'll be doing these. So I just thought it'd be good to see them a little bit before then. So if we think about your mean reaction time, that's 1 point along your distribution, typically around where the peak is. There's some caveats there we'll come back to in our last module. And the standard deviation, that's another parameter that tells you about kind of how wide your distribution is. So we often, for simplicity, look at mean and standard deviation, but we can, with some kind of special math, and we have to have a bunch of trials, we can plot your distributions. So the idea here is that, and this is across everyone, that reaction time, if it's a compatible trial, was typically between 250 and 700 milliseconds. And the higher this is, the more common your reaction time is of that latency. So typically, the most common reaction time would be about 325 milliseconds. That's the compatible, the ipsilateral condition. When it's incompatible, it's harder, or the contralateral mapping. You can see reaction time shifts over, so it gets longer. The average reaction time, mean is probably more like 450 milliseconds. We can see that it's wider, so the standard deviation must be bigger. There's lots of trials that overlap between these two distributions. They're not completely separate. So if I didn't know which block you were doing and we got a reaction time of say 400, it would be hard for me to guess, well, is that an ipsilateral condition or a contralateral condition? Well, I don't know because 400 could be either. But if we get a really fast one over here, it's way more likely that it's from the compatible block. Actually, even if it's here, it's possible it's from the contralateral block, but it's way more likely it's from the ipsilateral block. And conversely, if you get a reaction time, say here, well, it might be from this condition, but it's way more likely it's from the green incompatible contralateral condition. Any questions on the group results? So just to see where we're heading for the end of this module, the fourth assignment, nothing drastically different, but now it's a results section, which we have a lot of practice with, and the new section is a discussion section. On later slides, we'll get very specific details on this. Again, individual or group, same group as before, different groups, either is fine. And we've got 3 weeks, so it's kind of about the same time frame as our previous assignment. We have one more module after this, and that one has a longer time frame because you'll do all sections. We also have Easter kind of sneaking in there, so it's bigger and you'll have more time to work on it. And because I always forget, I made sure to put the assignment upload already on Blackboard. Oh, here's our little calendar. So the colors are just showing the different months. And so a change to our schedule is that we're going to meet next Monday for a little data processing party. So the goal this week is to go run the flanker experiment in the lab and get your data, and then come to class on Monday ready to process your data. So I'll kind of demonstrate, hey, this is how it's done. But then, we can do it step by step at a time together, and I can check your data. We can make sure everything is perfect. And then, so by the end of this class, you'll have your graphs, and then you'll need to, of course, write the results section to describe what you're seeing. And then the new section is the discussion section. Questions on timelines? It's pretty normal for us. Okay, so the bulk of today's class is going to be on what do we know about the flanker task, what have cognitive psychologists learned, and then also how to write a discussion section. Well, let's talk about the flanker task first. And we'll start with the paper, but then get into other aspects of the flanker task. Yeah, so here's kind of our topics for this section. We'll talk about the Erikson flanker task. I think instead of calling it Erikson, we just refer to it as the Erikson flanker task, hopefully acknowledging both authors. Then we'll get to the modified flanker task. That's the one we're going to use. It is very famously used in the attentional network task. So we'll talk about what that is. And then we'll talk about a model that explains the modified flanker task, the original flanker task, even things like Stroop, stimulus response compatibility. It's the dimensional overlap model. And it's kind of like the dual processing model, but a little different. So the Erikson and Erikson flanker task study, the one that I had you all read. So just a little background. So Barbara and Eric, so Charles Erikson went by Eric, which I think that's because Charles isn't, people don't use Eric or Charles interchangeably, do they? No, it's not one of those names. So I think he went by Eric because it was in his last name. So at University of Illinois, Barbara was a research associate there, so she just did research. And Eric was a professor, so I'm assuming he taught classes and did research. And I'm not sure when in their career they were married. I don't know if it was prior to Illinois or they met at Illinois, so I don't know that. But they were part of the greatest generation, and that's so Eric. I guess Eric served in World War II. He landed in Europe 1 month after D-Day and was involved in active combat. So that's where the greatest generation comes from. Eric was famous even during even when he was alive. Sometimes the research doesn't kind of get famous till after you die, kind of like artists. But Eric was known in his time even for this experiment. He was the editor of Perception and Psychophysics from 1971 to 1993, which is when he retired. And that journal was eventually renamed to Attention, Perception, and Psychophysics. I forgot to look up the impact factor of that journal, but it is a fairly fancy one. So this experiment itself, what they wanted to look at was the process of visual search. And I can't remember why, but I was telling my kids about this the other day, and they are almost 10 and 5, and they have a game where they like to spot yellow cars, and whoever sees it first gets to punch the other one. Thankfully, they don't punch each other hard. And I was like, oh, we're talking about this in class. So, you're searching the environment, the target is a yellow car, and there's lots of distractors. So, all the other cars are cars, but they're not yellow. And that's kind of what they wanted to look at too, the idea of looking through your environment for a target that's filled with distractors. And another good example of that, I'm not sure if my children know about this one though, is where's Waldo? So you've got a scene, you're searching it, the target is Waldo, and then there's all sorts of distractors. So there's people that could distract you, but we know Waldo I hope he's not cropped out, wears, red and white hat. So anything red might kind of be a bit of a distractor as well. And Waldo, thankfully, is visible here. I remember where he is, but he's hiding right here. Of course, they make it very hard, and they don't have his entire body visible. So you can imagine that this is hard, finding a target amongst distractors, and then we can do things like, well, what if we make the distractors more distracting, or more relevant, or in other words, more similar to the target? So now imagine trying to find Waldo when everyone kind of looks like Waldo. And here, I think I'm gonna have to go back to the front page to see, okay, what exactly does Waldo look like? So he's gotta have a cane and glasses, but some of these, I don't know why they're not Waldo, but the real Waldo is here. Probably silly things like the number of stripes on his shirt. So I think this is relevant, although we're going to look at it very much in terms of how does the mind do this, and we won't worry so much about humans in the real world doing this task. But there are researchers who look at that and the idea of identifying targets, a lifeguard identifying someone drowning amongst swimmers, so targets and distractors. Lots of military applications, of course, to this as well. So just a bit on the Ericsson experiments. I know you've read it, so we'll only look at a couple things. This is some of the equipment, so it's always kind of fun in these old studies to look at what was actually used. This is the tachistoscope, where they looked in this with their right eye. So nowadays, I was going to say you'd use a monitor, but you actually would use a modern tachistoscope. And the important thing about this is it ensured that the stimuli were all visible within your central visual field. So they're going to be presenting letters, but if you present letters like from here to here in someone's visual field, they can only see the middle ones clearly. If I want to see these ones clearly, I'd have to do saccadic eye movements. And typically they flash these up and they disappear and there's not enough time for that. So we want all the stimuli to be clearly visible. So we use a tachistoscope because it will control that they all appear in your central fixation. You would start the task. I can't remember if you pressed a button or released a button, but you'd say, okay, I'm ready for the trial. You'd be looking into this. This would control the timing of the stimuli. And this device I think this device is the one that will record your reaction time. And it was based on this lever, and you're either going to the left or the right, depending on whether you saw one target or the other one. So some fun old equipment. Probably easier to do nowadays, but we would still want to use a tachistoscope. They had lots of conditions in their experiment. 6 of them, and we're just going to focus on 3. So, oh, we're losing a letter there. So, once we get to the modified flanker task, it actually makes everything a bit easier. But in this task, they said if you saw that, sorry, there's an H hiding there, an H or a K, you would move that switch to the left. So there's 2 possible targets for a left response. There's also 2 possible targets for a right response, S or C. We're going to focus on conditions 1, 3, and 6. These are only one example of the stimuli. We'll look at more examples later. So if you get this, the key is these flanking letters, the 3 on the left and the 3 on the right, they don't matter. You're trying to identify the one in the middle. So here, okay, the middle one is H. Sorry, again, this should be H here. So when you see that, you should move the switch to the left. So we call that noise, same as target, because the noise, the 3 flankers are the same as the target. Condition 3, noise response incompatible, or we could say that, I'm not sure why the names jump around so much. You could call noise different as target, maybe. So here it's still an H, so you should be responding left. But the S, remember if you see S, you should switch the right, but only if the S is in the middle. So this is kind of like, those distracting Waldos. They're not actually Waldo because they're distracting you into thinking, oh, maybe I should make the wrong response. And then this is kind of control, right? We just give you an H or we give you an S or potentially a K or a C and you can respond left or right. So it's just how quickly can you see and identify a single letter. Any questions so far on this task? This work? That? So here, these are all trials where the H is the target. So you should respond left. So just an H, well, that's going to be pretty easy. A bunch of H's, we'll actually see what they found and what they predicted for this. But we can definitely say that this one's going to be harder. Because you've got the H, okay, I want to do left, but now all those flankers are saying, oh, are you sure you don't want to go right? And it's like, no, we want left. We need to avoid, we need to ignore those flankers. There could also be these trials. This is where the target is S, so you should be making a right response. And I guess there'd also be trials involving K or C, but I'm kind of ignoring those now. So just the target, or the target 7 times, or the target's flanked by letters that are suggesting the opposite response when you really just want to do what the middle one tells. So here are their results. Now, these are all 5 conditions, and we're really just focusing on 3 of them. So I'll just eliminate the other ones to make this a little more straightforward. And so that here they're calling this mixed control, but this would just be a block where on any given trial it could be any one of those letters, but just one at a time. So just imagine on this, H comes up, you respond left, or S comes up, you respond right. So this is kind of like a 4-choice reaction time. There's no flankers, and you're very quick at doing that. So your reaction time is the shortest. The other thing they added in is when they had 7 letters, how close were they together? So they could be 0.06 degrees apart, so very close, or 0.5, so a little further, or 1 degree apart. And I think one of the quiz questions was, why did they go with 0.06? Does anyone remember? Want to venture a guess? So that's what's used in typical text, right? So that's why they said, well, let's use this because that's normal, and then we'll do a wider and wider. So just to visualize what's happening here, So this is a trial where the center and the flankers are different. So it could be H and then S's or S and then H's, both kind of the same. They would average those together for the results. Here, all 7 are the same. Could be all H's, could be all S's. And then as the spacing gets bigger, we see reaction time decreases. And we'll talk about why they made that conclusion. Ooh. And then besides, so that's maybe the more subtle change. The big difference is whether all the letters are the same or if the flankers are different than the target. So you can see here your reaction time is quick, but when we have separate flankers, you're way slower. And that's a huge effect here for text that's spaced like normal text. And then the effect of the flankers decreases as the spacing increases. So what do they suggest with these results? And we'll look at their conclusions, and I'll try to explain them with some pictures, and then we'll get into, I guess, a deeper understanding of this task. So what they said first is that, well, visual attention is incapable of infinitely fine selection. So we can't just tell our brain, hey, all I care about is the center letter, so just focus on the center letter. Ignore those other ones. Which it would be good if we could, because then those flankers wouldn't have any influence on our behavior. But because we see this difference in reaction time when the flankers are the same or different, it means our brain can't ignore those. We can't just suppress them automatically. So that doesn't explain our visual attention, our spotlight of attention. What must be happening is there's some sort of minimal channel size that when we look at these letters, all of them are going to make it into the brain, and they're going to either facilitate or interfere with processing. So even though all we want to do is process the middle one, if this is our attentional field, we can't reduce it like I showed there. All of these are going to make it into your brain. And here, that's maybe not too bad, but if these are S's, well, then we got to go through more mental effort to say, well, we want to do the H response and not the S response. So let's look at a couple examples to explain that. So the idea here is where this is like the difficult condition or the incompatible, incongruent condition. And what has happened, they're close together like normal text. So all 7 of them make it into your mind. And that's trying to represent here. So all 7 of them are kind of need to be considered to determine which response we're going to make. So all 7 of these make it into your, well, let's say your working memory, and each one gets associated with a response. So for S, well, that means it should be a right response. So there's 6 of those, and then we have this one H, which is a left response. So that kind of automatically gets applied. We can't pick the one we want and then decide the response. They kind of all get mapped from stimulus to response. And then we need to say, kind of, okay, well, which one is the one we want? Well, it's the one that's in the middle, which is the H. So I've used green to show, hey, that's what we want to do. These red ones, these are distractors. These are flankers. These are bad. We don't want to do those. But it takes some, I think the actual quote was like available processing energy to inhibit these 7 and to excite that one middle one. So that's going to be slow because there's 6 things we don't want to do and 1 that we do. Now we're also, it does take some processing energy when all 7 are the same thing. So all 7 are the same thing, so we'd say, okay, for each of these we want to make a left response, but we still have to go through and say, well, we don't want to do these ones because those are the flankers, we want to do this one. So this will be easier because they're all suggesting a left response, but we still have to go through and inhibit these and excite that one. So that kind of explains, these results. All of those letters make it in, and it takes some processing energy to then map every stimulus to response and then to inhibit the response of the flankers. Now, why is reaction time shorter when the text spreads out further? Well, the idea is that, I'm trying to demonstrate it here, is as we spread the text out further, our spotlight of attention, all 7 letters no longer all make it into working memory. So here the idea is that within our attentional field, we see an S, an H, an S, and an S. So now there's only 4 things making it into working memory. So this is going to be easier. Instead of having to decide between 7 things, we only have to process 4. So for those 4, we're going to go from the stimulus to the response. So an H requires a left, S requires right, right, and right. And then now, it's kind of the same as before, but it's easier because there's only 4. And we have to inhibit these 3 and excite that one. So it's a similar story, but everything gets easier because the stimuli are wide enough that they're only some of them are within our central visual fixation. I think, yeah. So now all H's, I had to play with the size of the circle to kind of make this perfect. But imagine now, only four of the 7 make it in. So it's going to be easier to figure out the stimulus response mapping for all 4, and then to inhibit the 3 that we don't want. So that kind of explains, we still see a cost, but why it's smaller, because only 4 of the 7 are affecting us. And I don't know if they specifically guessed how many make it in, but maybe here when they're a little further, 3 make it in. So it'd be a little easier still. And eventually, if they're so widespread that only 1 makes it in, then the reaction time for both conditions should really be the same as this kind of control task. I guess based on the size of the visual field, you could maybe have done this experiment, like, okay, well, once they're 10 degrees apart, the only thing you're going to see is the middle one. Any questions about Erikson? Well, let's look at the modified flanker task, which is most commonly used. Sometimes you see like the Erikson kind of letters, and especially if you want to change little things. But very commonly nowadays we see the modified flanker task, and this is the one that we'll try. So 6 possible stimuli, and I don't know why we don't typically use 7, but it's typically 5 arrows. And it's always, you want to make the response based on the middle arrow. And I couldn't draw this great in PowerPoint, but these lines should be, exactly aligned with the arrow. So they're, lines without the arrow ends. So we refer to these as congruent conditions. So here the flankers are in the same direction as the target. So here, you want to make a left response, here you want to make a right response. And that's easier to do because, all arrows say left or all arrows say right. These are incongruent. So now these are going to make it into your processing, and you need to say, no, that's not what I want to do. I need to focus on the middle one and respond left. Over here, you would respond right. And these are neutral, which are somewhat of a control, where these lines will, still make it in, but they're not saying left or right. So they're going to be less distracting. You just need to focus on the middle and say, yep, I want to make a left response or a right response. And I guess to be clear that this is not the same as the Erikson and Erikson flanker task, we always call this one the modified flanker task. The modified flanker task it is used in different types of cognitive psychology, but it's had a pretty big resurgence with a new test that combines the flanker task, the modified flanker task, with kind of 2 other manipulations in cognitive psychology, and it's called the attentional network task, or sometimes shortened to ANT. And Michael Posner, huge kind of researcher in cognitive and neuropsychology. And I'm not exactly sure whether he was one of his students or what the relationship between Fan and Posner is. But I think Posner is the biggest author on that list. And often the senior author is last. So just to put this in context, I had to update these numbers because Ericsson and the Fan article typically get hundreds of citations each year. So these numbers went up even from 2 years ago. So Ericsson, I mean, has the advantage that it's been out for about 25 years more, but it's got like 10,000 citations. This Fan and All article, kind of the first one to demonstrate the attentional network task. And if anyone else talks about it or does a different version, they'll often cite the original. So they've got 5,000 citations, and then just to show you what normal citations you might get, and I guess this isn't even fair because this is my article with my highest citation. So 21 is, for me, I'm actually very impressed with. But it's nowhere near kind of the impact of these legendary studies. All right. So how does the attentional network task work? We won't be doing this in the lab, but I'll just show you what it's like. It's actually fairly hard. When I've done this as a participant, you gotta be like really focused on every trial. And when we do stimulus compatibility, it's like, oh, left, right, that's super easy. When it's cross, it's a little harder. This is like, okay, like lock in, strap in, this is gonna be tough. So you get a fixation, that's easy. And then on some trials, you'll get a cue. So here it cues you and it's saying, hey, on this trial, the stimuli is going to be in the up position. Sometimes it could also, hey, the stimuli is going to be in the down position. So then you know, okay, gotta look up, gotta look down. And sometimes it'll be 2 stars, and that means it could be up or down, so you don't actually know. Then it goes back to just the normal fixation, and the exact timelines are shown here. And then after a short period of time, your go signal will appear. And on this trial, it's going to be in the top because it told you in advance, hey, it's going to be up top. And we'll look at the different versions that this can be, but it's basically what we saw before. So this is an incongruent condition. Could be congruent, so all arrows the same way. Or it could be neutral, where there's, one is a middle arrow, and then we just have some lines beside it. So that appears, and then you make your response. It's a left or a right. I guess we could use the switch like Ericsson, but it's typically just keyboard. J or F? What keys do we use for this? F or J? Had the right keys, wrong order. And then you make a response, and then you get the fixation back, and then you restart for the next trial. So it doesn't look too bad, but the challenge is the different types of conditions. So the targets, it could be one of these 6. That's not too bad. That's basically the modified flanker task. But the other things they play with are the cue, and there's a third type of cue I didn't realize. So no cue I'm blocking it. Could be up, could be down. I'm not sure what that one means. Now, the difference in how fast you can respond when it's congruent versus incongruent, they refer to that as executive control, or the ability to resolve conflict. And by conflict, they mean in those flankers. So these arrows say, hey, go this way. That arrow says, hey, go that way. So there's a conflict that needs to be resolved. And the idea is your executive control parts of your brain resolve that. More on that later. The other thing that we're measuring in this task is alerting. So how helpful is it to get a cue? Slides here. They got the numbers right. So for alerting, the idea is when there's no cue, so if you just get no cue, then if you look at the timing, the go signal could be in 500 to 2,000 milliseconds. So you're like, okay, no cue, then you don't really know the timing. So you just have to wait for that go signal. When you get a double cue, although you don't know if it's going to be up or down, But it does tell you you're going to get the go signal in exactly 500 milliseconds. So you should be better because you can better anticipate when you're going to get the go signal. So that they call that benefit of alerting. You know more about the temporal sequence of events, or it decreases the variability of it. And then the last thing they measure is orienting, and that's a center cue, okay? You don't know, is it gonna be up or down? You don't know, versus an up cue or a down cue. So when you get an up cue or a down cue, you know, oh, I'm gonna look up and I'm gonna be better because I don't have to wait for the go signal and say, oh, where is it? There it is. So it's reducing the, the demands of making an eye movement up or down. You know, hey, I can focus up or down. So they call that orienting because your eyes are orienting to the proper position. So when you get a spatial cue, we expect your reaction time to be faster, and that's the benefit of orienting. So the neat thing about the attentional network task is that we can measure 3 types of attention. That's how they refer to it. So there's executive control, and I bolded it because that is the one we're focusing on because that is the effect of the flankers, congruent versus incongruent. And just in this original study, they found you were 84 milliseconds faster in the congruent condition. Or I guess, conversely, 84 milliseconds slower in the incongruent condition. Alerting, so how much faster are you when you know the timing of it? Well, you're 47 milliseconds faster. And what about orienting? When you know spatially where to look, you're 51 milliseconds faster. And the really neat thing about these 3 effects, or these 3 attentional network systems, is they've shown that they are distinct. Both in terms of, well, reaction time is 1, but if we look at this here, it's like, well, this looks bigger than these, but these don't really look any different. So they've looked deeper to prove that these are 3 different brain processes, or 3 attentional systems. And they've done that with EEG. So when they look at the timing, and we won't worry about what they're showing here, but they have different effects, so different brain activity timing. Then when we look at your anatomy, so when we use fMRI, we see different parts of the brain are showing advantages depending on the type of task. And finally, with the chemistry, they've actually shown different, what do we call these? I guess these are neurotransmitters? These all neurotransmitters? Different neurotransmitters are involved in these different tasks. So the reaction time itself is kind of just the tip of the iceberg. The deeper we look with these, and multiple levels with some very advanced technology, we see that the brain is benefiting from different mechanisms. And as you can imagine, I guess the complexity to that, the reliance on modern technology has made this a very popular tool. They've also done things, and I don't know the research off the top of my head, but they can look at a clinical condition, and they can test you on this, and if you're bad at this and not these, then it suggests, well, maybe you have damage to these brain areas, or to your norepinephrine system. All right, let's go specifically focus on the executive. So that's the flanker, modified flanker, congruent and incongruent effect. And you know how? Let's go back here. So we can take reaction time here, subtract them, and we get a difference of 84 milliseconds. We can also do that with fMRI. So fMRI shows us blood flow to the brain. Sometimes more blood flow can mean more activity. It gets a little complicated with training, because as you train, sometimes you see decreased blood flow, and that's actually, well, it's not that that area isn't active, but it's now more efficient. So it can be a little complicated. But what they do is they look at the brain activity when you get incongruent arrows versus congruent arrows. So lots of the areas of the brain would be active in both of those, but then they subtract them. So just like we do that for reaction time, we can do that for brain activity. And what they found, and it can be difficult to know, hey, what are we looking at here? But they found kind of 3 areas that are more active when it's an incongruent stimulus. So I get these right. Let me go with my notes. What's going on? I like to get my pages mixed up. Oops, too far, too far. All right. So when you get those conflicting flankers, the three brain areas active are the anterior cingulate cortex. We'll look at that next. Right and left prefrontal areas, and I'm kind of lump those into the dorsolateral prefrontal cortex. We'll talk about that a little bit too. And then also the left and right fusiform gyrus, which we won't worry about. So here's the anterior cingulate cortex. So maybe, well, I guess it would be kind of a deeper area. So maybe that area there, that's active. And the neat thing about this is the anterior cingulate cortex is involved in monitoring conflict. So, you get arrows pointing this way and that way, that's a conflict. And the ACC picks up on this and says, ooh, there's a conflict. It is then going to activate the dorsolateral prefrontal cortex to resolve the conflict. So it's very spread out behavior. The ACC says, ooh, a conflict, but it doesn't solve it. It just then notifies the dorsolateral prefrontal cortex. So if we go back here, that's probably what they're maybe showing over here. So it'd be more on the surface. These ones are more deeper regions. So, okay, there's a conflict. We've got arrows different ways. Then the dorsolateral prefrontal cortex would take over and have to resolve the conflict and somehow know that, well, we want to do the middle one and not the flankers. Any questions about Span and Posner's attentional network task? Well, let's segue into our dimensional overlap model that can explain the flanker task and lots of other tasks. So in Ericsson, when they talked about this sort of process, and when I was doing it with some hand waving as well, they talked about available processing energy. And okay, 7 things will take more energy. And I think that's like, okay, sounds valid. And then if one is in the opposite direction of the others, that's going to take more energy. But it's not a super detailed explanation. It's definitely not at the level of the attentional network task that gets into brain regions and even biochemistry. So one thing that's been done was to try to better model what's happening in this flanker task. And previously we looked at the dual process model, and it's really good at compatibility, but it's not very useful when you've got more than one stimulus. So remember here we've got the target and those flankers. We can't really shoehorn that into the dual process model, so that's why I crossed it out. And instead there's, I think a newer model, I think of this, when this was published, is the dimensional overlap model. So Professor Kornblum, Sylvian Kornblum, kind of a cool name. This is kind of his model, but there's lots of publication from him and his colleagues and then other groups as well. Kind of came out in the 90s. Those are kind of 2 of the key publications. I wonder if this website still exists. I think it might have gone down at one point. Let's just double check. Because it's actually super helpful in understanding it, because the research papers can be a bit complicated. And on his blog, he kind of walks through things in a really helpful way. I guess if it's not up, then at least like the Internet Archive, you can find it. I was surprised when it was still a few years ago, because Professor Kornblum, I'm pretty sure is retired, and I'm not sure whether he's still alive. So maybe that's, if you want to check out the Internet Archive, you should see it on there. All right. So the key to this model is it's going to consider relevant and irrelevant, the overlap between relevant and irrelevant stimuli. And it is perfectly adept at handling multiple stimuli. So if there's a target and flankers, it can handle that. There doesn't have to be multiple stimuli. We'll actually look at that example as well. So it's a very robust model, maybe more so than the dual process model. So this is Professor Kornblum's taxonomy of kind of reaction time tasks. And I'll explain this table because it definitely appears a bit complicated at first. And he's saying here there's 8 different types of reaction time tasks. The way they're differentiated is whether these 3 columns are they no or yes. So this first task is maybe the easiest because we get a no, no, no. And then you can see there's going to be 3 versions where there's a single yes. And then we've got 3 versions with 2 yeses. So yes, 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 or yes, yes. And then what I guess is the most complicated is when you've got 3 yeses. So it's really just the 8 combinations of yes or no in 3 spots. So what are these columns? They're saying, hey, is there relevant stimulus and response overlap? So is there something about the stimulus that's the same as the response? Yes or no? Is there irrelevant stimulus and response overlap? Yes or no? And is there relevant and irrelevant stimulus overlap? Yes or no? And we'll talk about these. There's many examples. So you can see here, we have stimulus response compatibility. So that was our last module. We have, I've read some papers where people mentioned the Simon task, although there's 2 types of Simon task. I'm unsure which one is the original, which one is standard. I think standard Simon is the one that's some of y'all probably wrote about with like the tones. I love, anyways, let's not get into that. There's the Erikson-Flanker task. This is potentially impossible. We don't have an example of it yet. This one's quite weird. And then down here we've got the Stroop task, the original Simon task, and the modified Flanker task. So these 2 Flanker tasks, one reason it's important to make this distinction is they do not share the same overlap. But let's look at, I think, 3 or 4 examples of this. And we're going to start with task number 2, stimulus response compatibility. So that would be the ipsilateral task and the contralateral task. So here's our ipsilateral mapping, right? Left goes with left, right goes with right. And let's look at it in terms of this taxonomy. So what is the relevant stimulus dimension? What are you paying attention to? Well, you're paying attention, is the stimulus on the left or right? And that's the critical thing of this task. Where is the stimulus, left or right? Are there irrelevant stimulus dimensions? No. So here, there's nothing else to the stimulus trying to distract you. Now, what's the response dimension? Well, the response is left or right. So this are the 3 things that describe the task. Now what we need to do to answer those 3 columns is we'll look at the overlap between them. So the first is there overlap between the relevant stimulus dimension and the response dimension? That should be the first column. Well, stimulus dimension is left or right, response dimension is left or right, so yes, they have overlap. It's the same thing, right? Left or right, left or right. So we say yes, there is overlap. For the next column, is there overlap between the irrelevant stimulus dimension and the response dimension? Well, this is none, this is left and right. You can't have overlap with something and nothing. So the answer is no. And then third, is there overlap between the relevant and irrelevant stimulus dimensions? Again, left and right, none, that's not the same. So we say no. So if we think of this task, and we could have done contralateral as well, you get the same thing. It is a yes, no, no task. Now let's go back to our table. Oh, here's contralateral. We'll come back to that in a second. So we were talking about number 2, and see it's a yes, no, no. So that's how we get to those yeses and nos. So here's contralateral, and although reaction time is going to be longer here, it still is a yes, no, no task. Any questions about applying Kornblum's taxonomy to stimulus response compatibility? Let's try another task. We're gonna go kind of easier to harder. We won't do every one. Okay, Erikson-Flanker. So think the S's and H's. All right, relevant stimulus dimension So the thing you're focusing on here is what is the middle letter. So middle letter. Irrelevant stimulus dimensions are those flanking letters, right? Now we have things that are irrelevant to the task, and that's all of our flanking letters. Response dimension, it's, I guess, left or right, either way. So first question, is there overlap between the relevant stimulus dimension and the response dimension? So I didn't add arrows here, but we've got relevant stimulus dimension and response dimension. Well, no. This is left or right, this is middle. Those are different things, so we say no. Is there overlap between a relevant stimulus dimension and the response dimension? Flanking letters, left or right? Yeah, I'm gonna go with no. But then lastly, is there overlap between the relevant and irrelevant stimulus dimensions? Yes, because they're both letters. So the original flanker task is a no, no, yes. So number 4, no, no, yes. I think we're gonna jump. Do we do Stroop first? Okay, we're gonna look at Stroop. I don't think we. No, that is not Stroop. Okay, well, it is a number 8. We're gonna do the modified flanker task, and I think after this we'll do Stroop. So this is gonna be similar, but not exactly the same as the Erikson flanker. So what's our relevant stimulus dimension? So what are you looking at to respond in this task? Middle arrow. Okay. So it's the middle arrow, and is it left or right? Now what's the irrelevant stimulus dimension? Which stimuli don't you care about? Flankers, right? Flanking left or right flanking arrows. And response dimension, what are you doing to make a response? It's a left or right response. So we've got lots of left and rights. So this is going to be a yes, yes, yes task. Because there's overlap. I think it's these two overlap, right? They're left or right. These two overlap, left or right, and these two overlap, left or right. So when you're doing this task, your brain has to deal with these 3 types of overlap. All the other tasks were easier because you only have one type of no overlap, that'd be the easiest. One type of overlap, it's a little harder. 2 types of overlap, a little harder. And then the hardest is 3 types of overlap. So we see that with the modified flanker task. We also see that with the Stroop task. And I don't think we tried this for ourselves, but we talked about it once before. So in the harder version of the Stroop task, the instructions are to say the color of the ink. So here, ideally, well, for the correct response is to say green, because the color of that ink is green. The problem is the brain automatically wants to read the word, which in this case, the word is blue. So you kind of have to suppress saying blue, that's kind of the automatic response, and say green, the ink that it's written in. So here, the relevant stimulus dimension is the color of the ink, or color of the text, sure. The irrelevant stimulus dimension is the actual word, and the response dimension, well, what you're trying to say is to say the color. So here, color is involved in all of them. So there's going to be overlap here, there's going to be overlap here, and there's going to be overlap here. So Stroop and the modified assignment task have a lot in common because they're both these yes, yes, yes tasks. There's a neat study, I'm trying to remember if it's also Posner, who compared the brain activity in the 2 tasks, and he showed both of them activated very similar areas. They both activated the anterior cingulate cortex, and they both activated the dorsolateral prefrontal cortex, but slightly different areas. So let's just say one activated it here, the other one activated it here. So they kind of said, well, they're very similar, but a tiny bit different. Any questions about this taxonomy? by Professor Kornblum. All right, so for self-experimentation, and we won't take too long here, especially because you won't be writing a method section, so you don't need to know the exact details, but we're going to be doing the modified flanker task. And I know we've said a lot of different flanker tasks, so let me just go back to the picture to show you all. So it will be like this. On each trial will be one of those 6 stimuli. These will be more aligned, and you'll be responding left or right with the F and J keys. What will that look like more precisely? And so it's, log into the computer, same as before. This involves 5 blocks. So the first block is just kind of a warm-up to get you used to the task. So just 30 trials, randomly those 6 stimuli. And this is all going to be considered practice, and we won't analyze practice later. But once you're warmed up, you'll do 4 more blocks. So everyone will do blocks 1, 2, 3, 4, 5 in order. And blocks 2 to 5 are like the test trials where we will keep your reaction times. And each one has 84 trials. And if you make a mistake, it will recycle the trial to the end. So don't try to count 84 as you do it. But if you make a mistake, then it will add like an 85th trial. What are considered bad trials? If your reaction time is shorter than 100, that's probably faster than humanly possible on this task. I think the computer will tell you. It'll say, wait for the go signal. If your reaction time is greater than 1,000, and often I like to think of that as distraction, it'll probably forget what it says. But it will recycle that. And then most commonly is an incorrect response, which, I mean, some people might make no mistakes. Maybe it'll be 5% on the harder trials. I guess I'll pull out those data after. You won't do that individually because it usually doesn't work out great on an individual level. Questions about the task itself? The other different thing from the previous ones, I didn't make a pretty splash screen. So when it starts, I think it'll just say like press enter to start. It'll be right into the task. It doesn't explain it, but it'll be the F and the J keys for left or right, and it'll be those 6 different stimuli in pretty quick succession. What are we going to extract from the data? Kind of like before, we're going to find out reaction time mean and reaction time standard deviation. And now we have 3 conditions. So you'll have to calculate these for neutral trials, congruent trials, and incongruent trials. There are 6 types of trials, but 2 of those get averaged together. So you can think of this as congruent left, and this is congruent right. So you'll have a bunch of these, but we'll average together all of those trials for our congruent score. Because we really don't predict anything different for a left response than a right response. Even for people who are right-hand dominant, maybe your right hand is like 1 to 4 milliseconds faster. But that size of effect, we would need to look at maybe 100 people and then average things to see it. Incongruent, you'll average all these trials together because, again, they both should be slow. And then neutral trials, what we typically see here is that neutral is typically either comparable to congruent or sometimes a little bit slower. So there's probably not enough of us to see that effect nicely, but these are typically fast, this is typically slow. So we're not going to worry too much about this because we'll go through this together on Monday. But these are the output from the experiment. So after you run all the blocks, you can then run the program, forgetting its name, that will put all your data together. It has Excel in the name. Sorry, I forgot. Hopefully it's obvious when you're in the lab. And it will actually just give you one file. It'll take all 5 blocks and give you one Excel file, which is maybe easier than last time. These are the columns, but we'll look at those on Monday. So I've opened up a file here and put in the headers. So it's fairly typical kind of format that we've seen already. But we'll talk about, how to sort it, what to remove, and we'll do this all together next week. Oh, these are the steps, but we won't worry about those now. And what you want to get eventually are your means for your 3 conditions and standard deviations for 3 conditions. And instead of making 2 separate plots, we're going to put them, all 6 of those data points together in one plot. We'll look at how to do this. If you had stats with me, you've seen this once before at least. But the bars, the blue bars here, those are your means. Just to reiterate that, did some little animations here. And then the size of these are the error bars, and they're the size of your standard deviation. And actually, I guess this would be 1 standard deviation above, and this is 1 standard deviation below the mean. Let's go to our table and see. Just trying to think. Well, I think this table is these data. So let's see if it makes sense. So neutral 367. Neutral 3, yeah, that could be it. And then the error bar is about 37. So it should go 37 up. So that's about 400. That makes sense? 360, yeah. Okay, so I think those go together. This is my data from 2 years ago. I did it again the other day, so I just wasn't sure if I had updated one and not the other, but I think they're both from 2 years ago. Okay. Any questions about doing the experiment? So the goal is do it this week, get your 1 giant data file, and then be ready in class with your laptop next week so we can process it. Let's take a quick sanity break before we talk about the manuscript. So it's 5:26. Let's start back up at 5:30.
આમાં જો વિન્ડોઝ પછી ઓપ્શન દેખાય ડેટા એક્સટ્રેક્ટ કરવાનું પછી કોઈ બધું કાઢવા માટે સોલ્વ એ કશું જ નથી આપણે તમારા ખૂબ અલગ છે આપણે ગયા સર્વિસ સારું ક્યાં છે જે લેપટોપ
What are we talking about?
stats.
Stats for Professor Lachlan?
So we were discussing about the last Excel thing.
Hmm?
She was not able to do it in her computer
Oh.
because it's Mac.
Yeah. Yeah. Which part would it not let you do?
Like the first one, data extraction.
Oh, okay. I was working with a student the other day on a Mac, and there is a way to do it, but you're right, he did not have the same. Because in here, I remember he showed me, but I forget now. So this is obviously a PC. So there's the data, get data, and it's not the same on Mac, but it is somewhere. You definitely find it next Monday. Try to remember if you can just do like file open and then say, hey, I wanna, no, that wasn't. There is some way. That was Joe actually, right? It's just like 3 days ago, I think he showed me that. We'll figure it out. All right, let's talk about your next paper. So the sections in that paper, you should have the title page, same as always, so you should be good at that. Results section, which we have some practice with. Discussion, that's the new part, so we'll talk more about that. And references, well, we've done that before too. So just so we have this in one spot, but you know, there's your title page. Results, we've seen this before, but I just wanted to reiterate. So we only need one figure per participant because the bars are the means and the error bars are the standard deviations. And then for your text, you know, briefly describe the visual pattern in each figure, and I'll get you more feedback about how you did on that on your most recent assignment this week. But in the ones I've created so far, the results sections have been really good. Okay, discussion, that's the new part, so I'm trying to give some guidelines on how to write a discussion section. It's maybe the most section that is up to the most style, I'm trying to think of the right words here. You see the most differences between authors, because there is a bit of artistry to it. So some people who are better writers than me, you know, have these really flowing discussion sections, whereas a method section, you know, it's kind of a recipe. Even if you're a really great writer, it's not that much different from mine. But here's some advice, kind of what to do in the different sections. So you start with the support of your original hypotheses, and we'll break these down. This could be one paragraph or more, at least one. Then you want to talk about the similarity of your results to other experiments who've done a similar experiment. Again, that could be one paragraph, or if there's a lot to go into, it could be more than one. Interpretation. So what do your results, you know, tell us? And this is kind of the main part of it. So here I expect at least a few paragraphs. And then typically the last paragraph or the end of the paper are kind of the implications of your experiment and the results. And this, at least one, but could be a few paragraphs. So what I'll present next is kind of APA guidance. How they describe each of these, first in a few sentences and then in a short paragraph. And I'll show you examples from the same paper of mine that we looked at last module as well. Okay, so the idea for these sections comes from the APA Journal Article Reporting Standards, which is abbreviated as JARS. This is freely available on the web if you ever want to look it up there. There's a lot going on here. This doesn't apply to every study, and I think for our kind of simple studies, it's really only these parts that apply. So this is kind of what I showed you on the previous slide, and now we kind of have, you know, one or two sentences describing these sections. So support of original hypothesis. Provide a statement of support or non-support for all hypotheses, whether primary or secondary, and, you know, well, make a distinction between primary and secondary hypotheses. So you might have a primary hypothesis about reaction time mean, and a secondary about reaction time standard deviation. And when you get to your first paragraph of the discussion, you'll say, oh, our hypotheses were correct, or this one was correct, this one was not, or they both were wrong. Similarity of results. Discuss similarities and differences between reported results and work of others. And that's where you would, you know, cite other studies that have done the same thing. So let's see, I guess you could refer to the Fan and Posner ANT task, but there are lots of experiments out there that have done the modified flanker task. And if you search modified flanker task, you'll probably get more studies than you want. So you can find an abstract that looks good. Interpretation, provide an interpretation of the results. I'll add some details about what that means. And then implications, discuss implications for future research programs or policy. The JARS actually comes back and gives you 7 different things you can talk about here, and I'll list those. And I'm going to tell you to maybe talk about 2. Don't do all 7. That would be a lot, but 2 is probably a good number. Or 3 if you really want. All right. Are there any questions before we get more specific on each of these?
Are you gonna want us to include like limitations or?
I think limitations is one possible discussion within implications. So you might. But you don't have to. Okay, support of original hypothesis. This is just do we have just one paragraph? Okay. So more than one sentence, but still pretty brief. Open the discussion section with a clear statement of support or non-support for all hypotheses, distinguished by primary and second hypotheses. In the case of ambiguous outcomes, explain why the results are judged as such. So if you predict A or B and you get C, that would be like an ambiguous outcome. But hopefully we all, replicate the typical flanker effect. We're going to look at one of my papers, which is a little more complicated. So hopefully it's still a useful example. But this is the first paragraph from one of my studies. And I actually spent a few sentences, and you can do this if you want, where I remind the reader, hey, what is our goal? What did we do? What were our hypotheses? Just kind of refresh the reader. And then finally, I get to, hey, were our hypotheses correct? So it's a little longer than just the minimum of jump right into your hypothesis. I'll just let you read that for a moment. So the part about, I think it's probably always a good idea to remind the reader of your hypotheses. So, hey, this is what we predicted, because that might have been way back in the intro, right? And now they've read the method, the results. So hold on, what were you predicting? And then this is kind of what APA was talking about, support or non-support of our hypotheses. We kind of had some complex hypotheses here, so we have some somewhat complex conclusions. We okay with the first paragraph? Similarity of results. This is APA's slightly longer description. Similarities and differences between your results and the work of others, where they exist, should be used to contextualize, confirm, and clarify your conclusions. Do not simply reformulate or repeat points already made. Each new statement should contribute to your interpretation and to readers' understanding of the problem. And I think what they're kind of saying here is you don't need to reiterate what you said in the results section. So if you say, hey, this is faster than this, well, they know that. So now moving forward, you say, hey, you know that thing we saw? Well, so did this group. Or, hey, this experiment, they didn't see it. So you don't need to rehash things that were in the results. Shall we see an example? I think I have an example. Okay, this is an example from that same paper, so you want to give that a read. So here we're saying our results, didn't do this. There's kind of this curve we predicted, and we say, oh, Crossman, and we explain a little bit. They did show that, and this is kind of a big thing. So there's actually a few more paragraphs where we kind of go into it. But here we've brought up a difference between our results and another paper. Any questions about that? Okay, interpretation, so APA doesn't say too much else here, provide an interpretation of the results. Okay, now let me try to explain that in a different way. So in the results, you're talking about reaction time, mean standard deviation. That's the results. The interpretation means we need to transition from results, reaction time, to what does that mean? And reaction time, that's what we're quantifying, but remember what we're inferring is information processing, cognitive psychology. What is happening in the mind? So in the discussion, the way we interpret is we use cognitive psychology theory. And here, in the results section, reaction time mean. So for example, this I would predict this, reaction time mean was longer in the incongruent condition than the congruent condition. That's perfectly acceptable thing to say in the results section if your data support that. In the discussion section, we want to interpret that change in reaction time. So here we go from talking about reaction time to talking about information processing theory. And the model that I encourage you to use is the dimensional overlap model. So yes, there's this reaction time difference, but now we can use the dimensional overlap model to say, well, the reason we have that reaction time difference is because there's conflict between all three aspects of this task. Remember, it's a yes, yes, yes task. So in the discussion section, you're not walking through the dimensional overlap model. You're just saying reaction time is fast, it's slow. When we get to the discussion section, that's where we use our cognitive psychology theory to interpret the change in reaction time. Now I have an example from my paper that's not exactly, a reaction time example would be better, but I tried to go with the same paper to be consistent. So let's take a look at this here. Maybe not my favorite example. So maybe I should give some background to this one. So in this study, so here we're talking about online control. And there's 4 kind of evidence for online control. Now, online control, you can't directly measure. So we measured all these things about the kinematics, how they're moving. Let's pretend reaction time was one of them. But then we're using reaction time to infer this cortical process of online control. And that's similar to how y'all will use reaction time to infer this overlap in the dimensional overlap model. So in the discussion section, what it means by interpretation is to transition from your dependent variable, hey, we measured reaction time, to what does that dependent variable mean? Well, it's referring to overlap in the dimensional overlap model and overlap between stimuli and response features. Yeah, it wasn't a great reaction time study, so we didn't have that there. This is study implications, and APA happens to have the longest sort of advice for this section. And there are 7 things in here. I don't know why they do it this way, but here's 4. And there's another 3 in this paragraph. So consider returning to a discussion of why the problem is important. So that's 1. What larger issue, meaning those that transcend the particulars of the subfield, might hinge on the findings? 2. What propositions are confirmed or disconfirmed by the extrapolation of these findings to such overarching issues? That's 3. And then there's 4 in this proper list. And so what I'd like you to do is find 2 of those that seem to be more relevant or more interesting to you and discuss those in your last paragraph or paragraphs. I guess you know what, limitations is not one of the options here. I think that is in some of the stuff I blacked out or redacted. That's the fun word. Let's see here. Oh, Wonder if it would fall under generalized
Contextual
ability.
issues.
What's that?
Yeah, the other contextual issues.
Ecological, yeah. It is something, but we won't worry about it. If you wanna put it in your paper, that's fine.
I don't have to, I'm not going.
I know in like lab reports in chemistry and biology, it was always like, what are the limitations? And I don't know, sometimes it's a bit silly. Even in published articles, you'll see some people do limitations and some don't. Some are very keen, like it should always be there. I'm always like, don't shoot yourself in the foot. And I mean, you don't want to hide something. You want to be like, oh, by the way, we didn't. But it's like, if you do a study and you say, oh, we tested all males, well, the limitation is then obviously that you didn't test any females. So, I don't know. I like to see it in, say, a thesis or dissertation, so that you know that the student understands, but once you get to the prose, professional published paper, then I'm like, I don't see this relevant. Anyway, it's my 2 cents. Okay, where are we here? Oh, sorry, we didn't see this yet. So if you want to read, oh no, sorry, let me go back. So implications, I actually had quite a few in this paper. There were actually 2 subsections. One was called Suggestions for Future Studies on Fitts Law. One called Suggestions for Future Studies on Limb Target Control. I'm trying to think if this article is on Blackboard. If you want to read it, you can see. But then in the last paragraph, there were some implications as well, so I included that paragraph here. And then the last part is your reference list, which I won't bother. I just put it here, so if you need this slide, there it is. But y'all have done this already, so you should be good at it. Bless you. Thank you. Any questions? So remember, our goal is, Elder, to get your data this week. We'll meet in class next week, probably not for 2.5 hours, but just to process your data, and I can also help with graphs and anything like that. All right, thanks everyone.